So traditional approach for small cell lung cancer has been one size fits all. I think gradually we're trying to shift to a subtype-specific approach, as I mentioned, based on the different RNA signatures. Small cell lung cancer can be stratified into different subtypes, which may have different therapeutic vulnerabilities as far as different treatments are concerned. And this figure is from one of our recent papers in Nature Reviews, and what it highlights is that on the left, you see small cell lung cancer that transforms from lung adenocarcinomas, especially EGFR lung cancers or other lung adenocarcinomas where they have a loss of function of these two mutations called TP53RB1, and then upregulation of some other mechanisms within the tumor, such as the PI3K pathway, Notch signaling pathway, which results in transformed small cell cells. More importantly, on the other side, on the right-hand side, you see this figure that shows different transcriptional subtypes and biological differences for these different transcriptional subtypes. So you have neuroendocrine high small cell lung cancer, which is usually the ASCL1 subtype based on the RNA signature. And then you have the NeuroD1, POW2, F3, not shown here, and the last one being the YAP high. The YAP high being also known as small cell inflamed. And this subtype tends to have higher presence of antigen presentation, higher checkpoint PD-L1 presence. And based on data that we've seen from the IMPAR 133 trial or the Caspian trial, the sub-analysis has shown that the YAP1 subtype does tend to be that enriched phenotype that has a longer and a better benefit to immunotherapy plus chemotherapy, likely because of the inflamed tumor microenvironment or the presence of more immunological signaling in this subtype. Now, based on what we have understood that small cell lung cancer can be stratified into different subtypes, there is an interesting upcoming study that is being led by Dr. Ann Cheng from Yale. It's called a PRISM study. And what they're doing in this study is they are subtyping small cell lung cancer upfront. This is extensive stage small cell lung cancer based on immunohistochemical markers into this subtype A, ASCL1, or subtype N, or subtype P, and then randomizing one cohort to immunotherapy plus a PARP inhibitor like olaparib, which targets the DNA repair pathway, especially BRCA1, BRCA2 related genetic alteration pathways, or subtype A and N, as is shown here in the figure, randomizing to an immunotherapy plus an ATR inhibitor, which is another type of DNA damaging agent, versus immunotherapy alone. And the third subtype being subtype I, or inflamed, randomizing to immunotherapy plus natural killer cell activator versus immunotherapy alone. So I think this is going to be one of those interesting biomarker studies that will help us understand how these different subtypes respond to different types of combination approaches, or whether these combination approaches actually do make a difference in the bigger picture.